we're going to discuss about the hormone structure, synthesis, and secretion. So, what are hormones? A hormone is chemical message transmitted in the blood that is secreted by an endocrine gland. Hormones can be classified into three categories, which is the amines, peptide and proteins, and steroids. Polypeptide hormones are proteins that act as a hormone. Size of the polypeptide varies from 300 to 200 amino acid residues. Cannot pass through cell membranes due to their size and water-soluble nature. Protein hormones are the most numerous types, secreted by many glands. Synthesis of polypeptide hormones is that polypeptide hormone synthesized in the same manner as any other proteins. DNA in the nucleus is transcribed to mRNA and translated into the protein by ribosomes. Protein is then processed by the Golgi apparatus and stored in the secretory granules. Many hormones undergo changes in the Golgi apparatus, secretory granules, like cleavage reaction, addition of carbohydrates group. The secretion of peptide hormone is that secretory granules released by exocytosis. This occurs when the membrane of the granule fuses with the membrane of the cell, causing the contents to be ejected. The process is triggered by calcium entering the cell. Polypeptide hormone releasing is controlled mainly by regulating secretion rather than synthesis. Now let's talk about the steroid hormones. They are small and fat-soluble, can pass through cell membranes, but they must circulate bound to plasma proteins since they are insoluble in blood, secreted by certain glands. Synthesis of steroid hormones derived from cholesterol. Cholesterol is acquired from the diet or synthesized within cells. All steroids have the same basic structure formed by 4 rings of carbon. Individual steroid hormones differ primarily. The process of the hormones is this: cholesterol, pregnenolone to progesterone, which the androgen is the testosterone, estrogens, e.g. estradiol, where the glucocorticoids is the cortisol. The mineralocorticoids comes from the aldosterone. The secretion of steroid hormones release immediately, so the rate of release is determined by the rate of synthesis, especially the synthesis of pregnenolone. Amine hormones formed by altering the structure of amino acids, secreted by the thyroid gland, adrenal medulla, hypothalamus, and pineal gland. Synthesis of amine hormones is that synthesized from 2 amino acids, the tyrosine, which is catecholamines or noradrenaline and adrenaline, and the tryptophan, the precursor of melatonin. The secretion of amine hormones also is that the granules are released by exocytosis. Rate of release is regulated mainly by secretion. Transport of hormones. All hormones secreted from endocrine glands are transported in the blood. Steroid hormones and thyroid hormones are lipid solubles and require a carrier protein to be transported in the blood in order to reach their target cell. Non-steroid hormones tend to be water-soluble, and as such they dissolve in the blood plasma where they are transported to their target cells. Mechanism of hormone action. The mechanism involves in steroid and thyroid hormone action. Endocrine gland secretes a steroid hormone. For example, hormone enter target cell by diffusing through the cell membrane and enter the nucleus. The hormone then bind to the receptor molecule. The hormone protein complex then binds to DNA and promotes the synthesis of mRNA. mRNA leaves the nucleus and enters the cytoplasm where it is translated to protein molecules. The mechanism involved in non-steroid hormones action is that endocrine gland secretes a non-steroid hormone like insulin, for example. The hormone is carried to its target cell by the blood plasma. The hormone then binds to its receptor site on the membrane of its target cell. The hormone receptor binding activates adenylate cyclase via a G protein. Adenylate cyclase then causes ATP molecule to be converted into cyclic AMP or C-CAMP molecule. The CAMP then activates various protein kinases which phosphorylates various protein substrates. The activated protein substrates induce changes in metabolic processes. Cellular changes are recognized as the hormone response. That will be it.